Was 55 years, so I don't Worcester. Oh, right. So it actually, I used to see it from the night. Yeah, but pretty much like you want your green cancer, you can't see it from the highway and you're letting out your mosquitoes. It's so really that you pass by. Oh, it's like yeah, there's yes, but it's a little short. Like, yeah. At the same because you can pass by and see holes across. Yeah. Ah, there we go. Yeah. I have one. I don't know how to do it. You know how to do it? I think whatever's explained. Oh, the turn on the light. Oh, oh, see, it says. Thank you. It's really not. Oh, that's all right. It's a lot.
Okay, I think we're going to get started. It's my okay, great. Yeah. Okay, so good morning and welcome everyone. My name is Lisa Olson, professor of biomedical engineering and auditory biophysics here at Columbia. Before I introduce our speaker, I have a few instructions to give. Today, we offer this lecture in a hybrid format with both a virtual and a live audience. Our speaker will present for approximately 40 to 45 minutes, and we will leave 15 minutes at the end for Q&A. For our virtual audience, throughout the presentation, please use the Q&A feature at the bottom of the screen, not the chat, to submit your questions. We'll try to answer as many questions as possible during the Q&A session at the end. For our live audience, you will have the chance to ask your question during the Q&A session too. So getting on to today's speaker, it's my honor and pleasure to welcome Catherine Whittington, Assistant Professor of Biomedical Engineering at WPI. Her research group uses biomaterials to capture dynamic physical changes in tissue microenvironments surrounding pancreatic tumors and lymphatic vasculature. Dr. Whittington's research is funded by the Pancreatic Cancer Action Network Career Development Award, Genentech Research Award, National Science Foundation, and the National Cancer Institute of NIH. Dr. Whittington completed a postdoctoral research fellowship in cancer cell signaling at Eli Lilly after receiving her PhD in BME from Purdue University. She also holds a BS in biomedical engineering from Louisiana Tech University. So let's give a warm welcome to Professor Whittington.
All right, Lisa, thank you very much for that introduction. All right, so I'll talk today about the work we're doing in designing more dynamic in vitro platforms. For pancreatic cancer lymphatics, and I'll talk just like one slide that kind of shows a couple of the other things we're doing, because I want to make sure that the students get recognized for the work that they're doing that's not exactly in this space. I put my contact information and some Twitter information here, but it's also on my acknowledgement slide. All right, so I just want to acknowledge the people who work in my lab. We definitely need to update because we've had some turnover, but just want to acknowledge the lab. The students named us the Dynamite Lab, so dynamic microenvironment tissue engineering, to kind of capture all the things we're doing, both in the disease modeling space, and also we have interest in the regenerative medicine area. So first, why are we interested in tumors and lymphatics? So I'm interested in how tumors actually invade in fibrotic cancer. So pancreatic cancer is a cancer that has a lot of fibrosis, and I'll talk more about that. But it does make the environment different from some other cancers that may not have as much of that extracellular matrix interaction. So thinking about how they invade in these cases and how that impacts what the cells are doing. Also, too, thinking about upon that invasion, they're not invading just into tissues, they're also invading into the vasculature, both blood and lymphatics. In pancreatic cancer, the lymphatics is a mode of metastasis. And so thinking about how the dynamic fibrotic environment is also impacting the way those vessels and behave on their own, but also interact with tumor and immune cells. And then also thinking about how fibrosis is part of initiation events, particularly around malignant transformation of pancreatic cancer with certain risk factors that are present. So why pancreatic cancer? So I got into or exposed to pancreatic cancer during my postdoc experience at Eli Lilly and Company. But really, we're looking at the fact that this is a cancer, and a lot of people are familiar with the fact that when people are diagnosed, they usually are not given a lot of options in regards to treatments. The outlook is not that great. And part of that is because when people are diagnosed, they are usually diagnosed at a late stage where there's already metastasis, at least to the lymph nodes. And so at that point, you have, if you look at this pie chart, you have over 80% of people diagnosed with at least metastasis to the lymph node, if not to distant organs. And when we look at the 5-year relative survival, going from the more localized contained tumors that are eligible for surgical removal, and look at how that percentage drops as soon as you get to a regional lymph node metastasis, and then even further with a distant metastasis. This really contributes to an overall survival rate for patients that's about 11.5%. That number has actually gone up, but one of the reasons why we're not seeing, we're seeing the increase or the elevation of pancreatic cancer as being responsible for cancer-related deaths, it's not because more people are getting it, it's because we're getting better at treating other cancers, and we're still pretty stagnant in pancreatic cancer. Also, there are significant disparities associated with pancreatic cancer in regards to Black Americans being more likely to have higher incidences of pancreatic cancer and mortality compared to their white American counterparts. So we do need to think about how we're looking at effective in vitro models that can be part of the preclinical modeling process so that we can understand more about what's happening with pancreatic cancer and how we can work to develop better treatments. So when I got into this as a postdoc, I was still working with people at Purdue. And so we were thinking about the way in which we're looking at this event of metastasis, the epithelial to mesenchymal transition that cells have when they break away from the primary tumor, start breaking down the matrix that's containing it, the basement membrane, and then starting to invade into those interstitial spaces, so that surrounding extracellular matrix and then onto the vasculature. And so when I was at Lilly, I was interested in the fundamental questions, but then also too, how could we integrate these things into more higher throughput screening approaches? And so working with the group at Purdue, we were looking at this tumor tissue invasion model that very similar to spheroid-based models where you have just an aggregate of cells assembling and then you embed them into a surrounding matrix. In this case, we have a matrix in both places, both in that aggregate as well as the surrounding, and we could dictate the properties of either of them to start mimicking different things happening around the tumor. So it gives us this customization, but also this radial invasion that is more similar to what we will see in the body rather than looking at something on a flat surface and looking for migration type events. And so this was work done with Purdue. TJ and Zhao were some of the main people that were on this particular study. This is in the lab of Sherry Harbin at Purdue. And so when we're looking here, this was a model that was set up so that we could also integrate this into higher throughput screening with automated imaging. So, this was part of being able to kind of develop a way to place this. But what we were able to see is when we look at, and let me change my pointer over. When we are looking at a tumor matrix stiffness, so the, the stiffness of the extracellular matrix material, in this case, it was type 1 collagen that is surrounding these pancreatic cancer cells in that aggregate compared to the type 1 collagen that was surrounding in the matrix. So tumor matrix stiffness is across the top, and then we have two different stiffness levels of surrounding matrix. We were seeing differences in how many cells and how they were moving away from the boundary of that aggregate into the extracellular space, with seeing that as we would expect, if you have more material, stiffer material, there's going to be fewer cells that are getting out from that tumor, but also too, it seems as though we were seeing differences in what the stiffness of the tumor itself was. And so in this case, we're looking at one cell type here, but when we looked at two different cell types of pancreatic cancer, one that has more mesenchymal, more invasive potential with the PANC1 versus the BXPC3, which is more epithelial, more cell-cell associated. We were able to see differences in the invasion patterns. So definitely more single cell invasion with the PANC1 versus more aggregate or collective invasion with the BXPC3. And then we're staining for markers of more epithelial markers in yellow with the E-cadherin versus the mesenchymal markers with the Vimentin in red, seeing that as we expect, PANC1 would be expressing the Vimentin, not so much E-cadherin, but we see a mix of E-cadherin and Vimentin expression in these BXPC3s. And we had previous work where we saw that transition of a lower to higher stiffness, where we were seeing a little bit more of that transition in those BXPC3 cells. So we're able to use this to look at different invasion patterns. And then moving forward with primary cancer cells, seeing that whenever the primary cancer cells were here in the top, we're not seeing really any invasion until we put in these cancer-associated fibroblasts. And also looking at in white, we see the extracellular matrix and how it is also being remodeled as the cells are invading. And then there are also physical interactions between those cancer-associated fibroblasts and the pancreatic cancer cells when we put them together. And so this was giving us an idea that this particular platform was able to pick up on some of these different types of features, depending on how we design the system. And so when I had my first undergraduate student working in the lab right after I started, we kind of started to go in this direction, and we kind of switched directions, but definitely want to get back. But I started looking and saying, okay, in that tumor sphere, what type of influence would having different matrix materials have? So we took it and had either collagen 1, or we put in more basement membrane related proteins into that portion of the matrix that was encompassing the spheres or the cells. And then we were actually not working with pancreatic cancer 1. This is, like I said, very early. We didn't even get on the confocal at that time. We were just on the fluorescent microscope. We were looking at U87MG, so a glioblastoma line. But seeing that even when we were keeping the collagen around it the same, the material that the cells were exposed to just from the tumor was making a difference. And some of these different patterns that we were seeing, where we have some more elongation in certain areas, some tighter invasion, not going as far. And so just seeing that both sides of it can be influential on what's going on with the cells. But we actually had to switch gears from that because I had one undergraduate student working with me at the time. Classes started, and so this was a bit more of an undertaking. So I hope to get back to looking at that work. But when we're thinking about what's actually happening in that environment of the pancreatic tumor, we have to really think about what's happening in the body. And so when we look at these sections of normal pancreas versus the pancreatic tumor, we see differences in the organization of the tissue. There's a lot of extracellular matrix that's being remodeled. There's excess deposition of the extracellular matrix. There's cross-linking that's occurring. Fibrils are linearizing and changing their orientation. And so this is increasing the overall stiffness of the extracellular matrix and making this a very heterogeneous environment, not just thinking about the cell composition, but also what's happening with the ECM organization. And so when we have this fibrotic activity, all of this chaos happening, it is impacting how cells in the pancreatic tumor are progressing and responding, but also too, on the flip side of it, it's also inhibiting some of the effective drug penetration. We don't look at it from that standpoint right now, but it is kind of a multi-factor situation for these cancer cells. And so when we think deeper about what the composition is, we have collagen and hyaluronic acid. So these tissue sections are showing in the normal tissue, how you have collagen and hyaluronic acid, how they are distributed out. Both of them are showed in brown and it's different panels, but then we get into the tumor, there's a lot more of it and it's not nearly with the same type of organization. So this is again, thinking about how we model these systems and start to capture some of these different features that are going on. And when we look at patients with these Kaplan-Meier survival curves, present survivals on the y-axis and time for survival on the x-axis, seeing that the darker lines where you have lower collagen 1 or hyaluronic acid is indicative of better survival outcomes compared to the gray lines that are showing lower survival for the patients when they have these high amounts of collagen and hyaluronic acid. So again, thinking about how those extracellular matrix components are factoring into the situation. And so thinking about how existing models, how well they are incorporating some of these spatial events and their spatial temporal events too, because this is all happening in a progressive nature over time. So that's, you know, what's really motivating us to think about how we could be more dynamic with the environments that we're creating for these in vitro models. And so as we're showing just kind of the differences in basement membrane that's keeping the tumor contained, it's got its own composition. We move to where you start to see this malignant transformation where the cells are starting to invade into that surrounding tissue as that basement membrane breaks down. Then they are now exposed to collagen, hyaluronic acid, collagen type 3. And then as they get, you know, worse and worse and start to progress, you know, they're still interfacing with this material. And so, you know, we're looking at what that means for the organization of the environment. We're seeing that as you start to add in, things change. So we're looking here on the right-hand side of the screen where we took confocal reflection images of type 1 collagen alone on the top. And then when we started adding in either basement membrane materials or hyaluronic acid, how it does play a role in how organization is changing with time. But we're thinking right now, because in kind of looking at what was working for us, what not working for us, kind of getting the heterogeneity in a controlled way for the organization was more of a challenge than I think I originally appreciated. And so we decided, you know, to start to focus in more on the stiffness events that are happening with that excess deposition and the cross-linking. Other people have looked at using dynamic systems for different types of applications, some cancers, some not. And so just kind of looking at different work where in this case, they are taking collagen and sugar threose and adding them together and using the threose as a cross-linker to see how it's impacting cancer cell invasion. So using this method to facilitate cross-linking, changing over time. And the case here, they're using an alginate collagen mix where they're using the calcium chloride to facilitate cross-linking and the alginate along with the collagen and seeing the impact that it has on the fibroblasts, just with normal collagen versus early events of when the collagen and the alginate are combined versus over time when that cross-link is occurring, you're seeing this change that's happening to the cells as the cells are experiencing these stiffness changes over time. In this case, it's a dynamic situation where they're looking at a breast cancer line and doing a photo cross-linking in this case. So they have cells that are on top of a methacrylated hyaluronic acid with matrigel, and they're using photo cross-linking in this case to facilitate the cross-linking over time and looking at a spheroid response. And in this case, it's an enzymatic reaction with these gelatin PEG hydrogels and using that reaction to facilitate the stiffening. So there's lots of different ways that people are looking at having these dynamic environments. In some cases, they are using it to stiffen, but perhaps not monitoring things over time. They're looking at different outcomes, but we're thinking about, how we can have this controlled way to look at it in our pancreatic cancer models. And so we opted to right now look at photo cross-linking as a way to progressively stiffen, looking at a methacrylated form of type 1 collagen using a photo initiator that will allow us to have some dynamic control over the change in stiffness with time, to have those transitions, eventually wanting to bring in more of the heterogeneity as well. But then thinking about how we're kind of reflecting some of the stiffnesses that we see changing from a normal pancreatic tissue, and then going into kind of earlier stage events, early tumor formation, other risk factors, something like chronic pancreatitis, where it's an inflammatory environment that's facilitating some of the stiffening, and then going into what cancerous tumor tissue is like. So how can we kind of start to span that range and look for the response of these cells, instead of going with an approach of looking at a low stiffness versus a high stiffness, and kind of seeing what maybe could be missing by not having some of these temporal events incorporated. And so Brian Rolifson is a graduate student in the lab, and so he's been looking at some of the things around what's happening with some of these different photo initiators that we have available to us. So we have a methacrylated type 1 collagen. We're looking at 3 different photo initiators, in this case, Ergocure, LAP, and ruthenium sodium prosulfate. One of the things that we're looking at is, could we get out of the UV range for exposure to cells and look at some of the visible range? So that's why the LAP and ruthenium are something that we're exploring. But basically, this is a sweep of time looking on rheological testing, looking at the increase of the shear storage modulus, G prime, and showing an increase during just self-assembly time, where the collagen is just exposed to its neutralization solution, and 37 degrees, so that it'll self-assemble. And then we see these increased points where that's where light exposure actually is occurring, to show that there's increase. Now, we were getting a lot of variability in the stiffness changes, so trying to tighten that up a bit, but ultimately we were able to see some differences in the max stiffness that we could generate with the Ergocure compared to LAP or the ruthenium, and then looking at kind of the change, because also some of these were starting out at different stiffness values, and so seeing kind of the change in stiffness to get a dynamic range, but seeing that ruthenium was giving us not just a higher stiffness in the end, but then also too, when we're looking at different amounts of light pulse time. So these were pretty long on the right hand side. I think they were like 10-minute exposures, and these were without cells in them or anything like that, just to make sure we're maxing out. But whenever we were looking to see, okay, if we were to just do like shorter pulses, could we, like others have done, just have dictate a shorter amount of time for exposure and be able to control how stiff the material gets. And so we're able to get some really nice control with the ruthenium compared to some of the other ones. So we had originally started with LAP, and so a lot of our work so far has been with LAP, recognizing that it's given us a smaller dynamic range, but then looking to make some transitions to ruthenium in the future, based off of the work that Brian worked on, as well as Fatima, who's an undergraduate student who worked with us for a summer. One of the things that we have to be concerned about is viability of the cells with these photo initiators, and also the light exposure. So students Jess and Hannah were undergraduate students we're looking at was if we had sustained light versus pulse light. So they set up to where they were looking at different concentrations of LAP. The 2% LAP is what is actually the manufacturer recommendation, but kind of seeing what's happening with these PANC1 cells on just 2D tissue culture plastic, to see kind of what's happening with the sustained light versus the pulse light. And we were seeing kind of what we weren't expecting. We weren't expecting the cells to just die, but we weren't necessarily expecting it to look like the light was helping the cells from a viability standpoint. And seeing that in the 3D situation as well. And so when Jess looked at a control situation of just no cells, just LAP with the media, she was seeing that, we're actually getting some of these signal changes that we would not expect, because we're not expecting the metabolic activity, because we're looking at this with Alamar Blue. So it's looking at metabolic activity. So we weren't expecting to see any changes in metabolism if there were no cells there. So we're realizing that the reaction with the LAP and the light likely creating some free radicals that were then being interpreted as metabolic changes. And so we're like, we need to probably change our viability testing. So some other students started working and looking at, these were two students, Maddie and Tiffany. Maddie was with us for a summer. Tiffany has been with us longer, but started to look at calcium AM, ethinium homodimer, to look to, as that is an assessment tool of an image-based approach. And so seeing that without light and with light, how we could better distinguish what's happening from a live dead perspective, and getting things together in terms of how we're really setting up this experiment, but ultimately looking at that as a better approach for us to actually look at the viability compared to when we were trying to track proliferation with Alamar Blue, since it has this nice feature of being looked at over time. But when we're looking at 3D, also with the not Alamar Blue, with the calcium AM ethinium homodimer, seeing where when we have the no light situation, which is our control, so there's still photo initiated there, but no light exposure, versus a progressive situation where you have cells that are grown for X amount of time, then they're exposed to the light on the middle of their culture, and then so they experience a stiffening part of the way through is our progressive case, and then static is where they're increased in stiffness from the very start, and seeing kind of some of the differences that we see there in regards to viability, seeing that our cells are still viable, with that progressive nature, but starting to just make sure that we're not inadvertently already killing the cells while we're trying to go through this process. So this work is work that Athenia, who is a graduate student in the lab, and then Ellie is an undergraduate in the lab who's been working with PANC1 and BXPC3 cells. And then we've also been looking at normal pancreatic epithelial cells. So essentially what I just described in terms of the light exposure, this work was still done with LAP. So looking at what we can anticipate with the no light control stiffness levels compared to when we have the light exposure. And then instead I'm showing the PANC1 right now, but then Ellie has also done some work with the BXPC3, and then another student been working with the normal cells. But starting to look and see kind of what types of things can we start picking up on, what type of differences are appropriate. Because we're staining here on the left-hand side with F-actin with using phalloidin, and then nuclear stain with DAPI and shown in blue. And starting to see kind of what types of features are we able to pick up over time, and what type of outcomes are the best for us to assess what's going on. And so the H67 are normal pancreatic epithelials versus the PANC1, which are pancreatic cancer cell lines. And Athenia looked at a whole host of different morphological characteristics to start seeing if we are picking up on some of these events, and recognizing that some cases we may be getting like obvious morphological changes, but sometimes we may be getting more subtle or not at all. But then, having kind of a multi-pronged approach where we're starting to look at immunostaining of things like ecadherin, vimentin, or others. But in looking at some of the metrics that are related to circularity, and kind of seeing if those would be indicative of more aggressive or invasive behaviors that are more aligned with what we think about with mesenchymal phenotypes. We're not really seeing anything, and this is not shown, but we're not really seeing anything in regards to spreading events of the cells compared to, we are seeing some differences in regards to the progressive situation, which is shown on for each graph is shown on the left-hand side versus the right-hand side is a no light condition. But seeing some emerging differences that may be coming about with elongation that could be related to EMT events. Again, following that up with looking at some of the hallmark EMT markers, thinking about mutation status of these cells. So in the case of the H6C7 cells, they don't have the same type of mutation that the PANC1. Pancreatic cancer, often over 90% of the cancers have a KRAS mutation with KRAS overexpression. You also have P53 loss that you often see as well with pancreatic cancer. So thinking about where mutational status may also be factoring in some cases to some of the difference we're seeing, but we're also seeing a very broad distribution and likely from some phenotypic heterogeneity. And so having to tease into, how we're representing our result and thinking about, an aggregate of the information compared to looking more from a distribution standpoint on what type of heterogeneity we have with these cells. But when we're also looking at a comparison with the BXPC3 cells, if you remember the with the BXPC3 cells, seeing how they are a cell line that is initially more epithelial in nature compared to the PANC1, but looking and seeing over time where things might be changing a little bit more. And this is more preliminary when we were looking at this, but we're seeing some clustering that's happening in the PANC1 and the H67 compared to the BXPC3, that the BXPC3 seems to be changing a bit in their cell-cell associations, which is not unlike what I'd seen in previous work, where when we're stiffening the environment, those cells were becoming more mesenchymal in nature in terms of adopting that expression of Vimentin and maybe losing some of their E-cadherent expression. So thinking about how in some cases they are staying rounded, but also they may be losing some of those interactions. So again, trying to tease out some of the ways that we are interpreting what we're seeing in our cell systems. But work that we're ongoing, I don't really have in this case. So some of the work that I've just been talking about is related to this project, but we haven't brought in parts of this project quite yet to present. But we're looking at how pancreatic cancer initiation events are impacted by risk factor exposure. So when we look to see all of the different ways that risk factors are impacting pancreatic cancer, there's lots of different risk factors. And a lot of this is based off of more retrospective type studies, where you're looking at the kind of relationships of what happened and then looking back at what could have happened. And so there's not always much in terms of biological answers around what's actually, what about these risk factors are actually impacting this cancer initiation. So we're specifically looking at this project in particular at obesity and how obesity is impacting the development of pancreatic cancer, thinking about what the crosstalk is between normal pancreatic cancer cells, but then normal pancreatic cells, pancreatic cancer cells, and perhaps pancreatic epithelial cells that do harbor the mutations. And so how are they communicating back and forth with with adipose cells in pancreatic cancer development? And so we were thinking and looking into exosomes. So these extracellular vesicles that are secreted, they have a lot of different content in them. Could be nucleic acids, lipids, lots of different things that are within these exosomes, but are they providing some type of signaling that is impacted in some way with this kind of progressive fibrotic environment that is at kind of some of these earlier stages. And so Athenia has been working on this project. Jean-Luc is an undergraduate student who's working on the exosome work right now. And then Paul is a new student who's going to be getting going on this project, an undergraduate student. But, kind of thinking about what is that exosome crosstalk that's going on. And so it's known that pancreatic cancer exosomes have the ability to play roles in transformation of stromal populations, but also it has an ability to impact the metabolism of adipose cells. But then when we go and look at adipose-derived exosomes, there's established information about how their cargo can be much more pro-inflammatory, changing the phenotype in different cells, but then also impacting both on the migration and invasion side of it. That would be more related to metastatic events compared to changing the proliferative environment of the cells with those interactions. And so when we're thinking about how that is all impacted perhaps by the changing microenvironment, whether it is the amount of exosome production, the sensitivity of either cell type to the exosomes of the other. So understanding more about what's happening in those crosstalk events. And then, as I mentioned before, thinking about that interplay perhaps with the intrinsic genetic factors that are at play, as well as these extrinsic mechanical or microstructural, the microenvironmental changes that are happening during this process as well. So this work is underway. We're getting the adipose cells going, and so going to start doing the collection of the exosomes and characterizing them, and then starting to look and see how they're impacting either cell type. But I'm going to switch gears and talk about some of the work that we're doing in the lymphatic space. I had a lot of background from my PhD in blood vasculature, nothing in lymphatics, but then starting to see kind of where there were interactions with lymphatics with our pancreatic cancer work potentially, but then also thinking about lymphatics as on their own. And so when we think about lymphatics as playing an active role in the body in a lot of different ways, it was, you know, often for a long time thought that it was much more passive, but there's much more active roles that have been identified. And we're thinking about their roles in inflammation and the immune response. I'm interested in the metastatic involvement that relates with not just with the metastatic, but with the cancer side of it, what's happening on the immune cell side of it and how the immune cells are being impacted. And then also things that directly impact the lymphatics. So outside of cancer, something like secondary lymphedema, where there is vascular insufficiency and how that plays into, you know, how the lymphatics are responding to the environment from a regulation and a dysregulation. So how is the environment working for them and how is the environment not working for them? But thinking about it from a growth and function standpoint, because we know that mechanical inputs can affect lymphatics in different ways. There is a hierarchy of the vascular structure, and how is that changing so how are the mechanical inputs and the outcomes being impacted by the fact that you just have different vessels along this hierarchy going from capillaries all the way into larger collecting vessels closer to the lymph nodes. But there's been established information in regards to biomechanical inputs like flow, stretch, and pressure that we know outcomes about lymphatics, both how they impact the growth and then the function of the lymphatics to not just have lymph fluid go through the vasculature, but also how are things crossing the vascular wall, so the barrier integrity. But one of the things we wonder about is what about the influence of extracellular matrix stiffness, because we know that that's also changing both in development and it's also changing in disease, but that's an area that doesn't get as much attention relative to flow, stretch, and pressure. And so we think about how the lymphatic capillaries are attached directly to the extracellular matrix around it. They would be sensing some of these stiffness changes. And others have looked a little bit at some of the stiffness changes. So in this case, they are looking at lymphatic endothelial cells on a flat surface, what this group is, and looking as they're changing the thickness of a gel that's on top of a glass substrate. So by decreasing the thickness of the gel, they are increasing the stiffness of the underlying substrate and seeing how the morphology of the lymphatic endothelial cells is changing from this elongated structure to something that is much more spread out. Another group has looked at these on hyaluronic acid hydrogels, looking at a difference in stiffness from around 30 pascals to 900 pascals, seeing these lymphatic endothelial cells staying for their F-actin and nucleus and how the morphology of those cells are changing. They were also looking at sensitivity to VEGF-C, so that's a growth factor that is impacting lymphatic vascular growth, and seeing how the sensitivity of those cells to the growth factor is impacting their growth. But we're looking at a lot of the work is focused in on development, and part of that is when we're looking at stiffness, we're looking at stiffness values that are lower and in a range that is more on the developmental side compared to some of those stiffness levels that we're talking about that we see in disease. And so, where could we fit in perhaps with our dynamic approaches to be able to look at that more broadly? Also from a disease, I have to give a shout out to Brian for this figure that he made. But he, kind of looked at different types of dysregulation or disruption that you can see in lymphatic vasculature that are happening in different pathological conditions, all the way from, disrupted formation, halted completely, to where we're getting like an overabundance of sprouting, or discontinuous vasculature to where, that's changing the barrier integrity. And so, one of the things that he really emphasizes, he's been going through and looking at all of this, is the fact that, function and growth are both being impacted when we're thinking about, these changes that are happening. And from a fibrotic standpoint, what's happening in growth and function, but then how can we separate out growth from function because it's often conflated and it's not really separated out, and where some of these individual events are being impacted differentially based on the environment. And then being able to separate out the ECM stiffness from some of the effects that you would see from, fluid flow and fluid pressure, because we know that the cells are mechanosensitive in some way. And some of the other work that people have done to look at some of these stiffness events have shown that some of the same molecules are being regulated by stiffness and by something like flow, but we don't know really the contributions of them to know kind of what is dominating the narrative there, or how they're maybe cooperatively working together. So got several students that have worked on just different aspects of this. So Brian is leading this, but we have Gabe, Tony, and Eleni, who have been involved in different parts, whether it's been developing and working to fabricate the microfluidic chips versus doing some of the cell ECM work. They've been involved in different aspects of it, but using a chip design that was used for a blood endothelial application from Brendan Baker's group at Michigan, but taking that mold, making the chips with PDMS, and then going through and having this area here that would hold the hydrogel. Once we put these acupuncture needles through and add the collagen, in our case, around them, so that they then, once everything's solidified, you pull the needles out, they create the channels, and then you can dictate what you have, in either channel, whether it's cells in both channels, cells in one channel, to be able to start to look at some of these events. And so, this is showing an image of that chip and the hydrogel, the hydrogel portion with the channels that have been created. So actually some of these images, not the one on the right, but the one on the left, Brian just sent these to me yesterday. So we were super excited to see some of this, but he had done some initial feasibility at a different time where he was able to show that, yes, he could form the channels and could flow through some sort of fluid through, and being able to show the distinction between the two channels. But this is kind of a more recent one that he's demonstrated that. And then we were excited because then he sent me the images where he has seeded cells in it. And it's a process of being able to get the attachment right for the cells. And so when he sent this to me, I was just like, yes, we've got it there, because he had also done some work with this with a different cell line, which it's great, but at the same time we wanted to get it with the line that we're looking at, which is human dermal lymphatic endothelial cells. And so being able to, show that we, now we're at a point where we're seeding the channels with the cells, and then we'll start to look at what type of sprouting events are happening from it, to start looking at the impacts of growth and function. In this case, we're setting it up to where there is a channel that includes growth factors that would help stimulate the cells to sprout in the direction. So in him putting in that system, looking at how that fibrosis element is coming into and impacting, not just from stiffness overall, but thinking about how cells do have an opportunity to do some sprouting into a softer environment first, and then over time, as a disease increases in severity, you get that stiffening. And so being able to kind of interject that in between and see how well we are able to match up with what we're observing in vivo. Because one of the things that we looked whenever we're going through literature is oftentimes there is a disconnect between what we're seeing in vitro and in vivo, and in some of these cases with trying to have more predictive in vitro models. So his next steps are going to be to look at the sprouting as well as the function, again, separating out those two pieces, and then looking at what are the signaling mechanisms that are dictating some of these events. So, some of these have been established in other situations. So something like a YAP-TAZ is a mechanosensor that we see in a lot of different areas, but also thinking about, in this case, it's been related to growth, but how is it impacting function, or is it even, does it even matter in function? But being able to separate some of that information out. And then the last part of the lymphatics that I'll talk about is around how we are thinking about those trafficking events that's going on between the lymphatic vasculature, pancreatic cancer cells, and immune cells. And so as we are thinking about, particularly on the immune side of things, what's happening in regards to how cells like the dendritic cells and macrophages are actually being trafficked through, because I think it's easy for us to think about how the fluid is just moving through the vasculature and the cells are within the fluid, but it's not like everything's just kind of whooshing by. There are adhesion events that are going on between the endothelial cells as well as those cells that are moving through. And so how is the environment or the microenvironment around those lymphatic vessels changing the expression of adhesion molecules, the production of chemoattractants, and how is that changing how these cells, once they are entering into the vasculature, how they are moving through the vasculature. So we have a newer student who's rotating in the lab, Stephen, who's starting to work on elements of this project. He's starting out first looking at some of the characterization of the material itself. So this is also helpful for Brian's work. But looking at the collagen materials and just what are the transport properties through looking at both collagen with and without a fibronectin coating, because the lymphatic endothelial cells also like fibronectin. So they invade into these microenvironments that are collagen rich, but also they attach really well to fibronectin. So looking with and without coatings. This was looking at using, in this case, he was looking at 10 kilodalton fluorescently labeled dextran, and seeing in these early studies, not seeing any differences really with the permeability through. He's starting to put cells on top of it to see if there are differences impacted by a cell monolayer. One of the things that he thought about is that I need to check and see if the cells are actually taking up some of the dextran. It looks like there are. So that is our next phase of paying attention to that and accounting for those. But looking to start to use this Transwell model as a way to look at impacts on barrier integrity as we change the stiffening environment, similar to what we also hope to do when we're in the CHIP model, but being able to use this model system for that. Oh, I'm sorry about that. I hit the wrong button. Sorry, I need to exit out of this because I don't know what button I hit for that. Apologies. Okay. And so then related to this permeability through the matrix, we also need to be thinking about, well, what is the structure of the matrix? So last night at dinner, we were talking about how we're not in the lab. So this is the only thing that I can take credit for that I did was I took the confocal images. But looking at, getting down and quantifying what's happening from a microstructural standpoint when we are undergoing this photo cross-linking to see how that's also impacting transport. And then the very last thing, I need to rehide the meeting controls. The last thing that I just want to give a highlight for, again, is the projects that I didn't talk about, but students doing some great work. So Jojo is a master's student who has graduated from the lab, but Maddie's an undergraduate student. They were working on a project where they were looking at making these partial 3D constructs to look at the impact of pore size on bone cell behavior. Then Isabella is a high school student who approached me to work on some things around uterine fibroids. So she started working with it from a high school science fair project. And so it's continuing on, but how we can look to make some models of intramural fibroids to start. And then Brianna is a co-advised student with a colleague in my department. And so she's looking at scaffolding techniques for skeletal muscle regeneration. And then we're also looking at how we can, potentially integrate into the scaffolds, either some work about pancreatic cancer or some work on the lymphatic side of it. But I will just end to thank all of the supporters financially that we have, which has been great. It took a little bit of time to get there, but we got there. Students past and present, because some of the foundation of where we go with some of this has been worked on by a lot of undergraduate students have passed through the lab. And then contact information, including where I spend some of my free time on Twitter. And then I want to put out there that I am looking to hire a postdoctoral researcher, primarily on the pancreatic cancer projects, but, opportunities to work on the lymphatic side as well. So thank you very much for your time.
Can you stop sharing now? And then I just have to do some phonology.
Oh, okay. Yeah, well, I guess I did just grab a book. Oh, okay.
Okay, we're good.
Okay. Oh, he was
So,
able to take over the
just a real
sharing.
Quick, ending part here, and, okay, so. It was a wonderful talk. We'll get to the Q&A in a second. So just want to call everyone's attention to our next hybrid seminar next week, Friday, April 21st with Jean Fan of Johns Hopkins University. And remember to follow us on social media to receive the latest updates. And you'll see our information here, and you can always access the registration information on our website at bme.columbia.edu. So now we get to go to the Q&A, and there are a couple of questions from our virtual audience, but we like to start with live questions. If anybody, oh yeah, Kate.
No.
But when you were talking about the barrier integrity, you talked about the microenvironment. I was wondering, so like I know people themselves for like the blood vasculature, you see like IgG and then each molecule, probably decrease, decreasing the integrity. So does anything like that exist or I guess, is there any equivalent to the lymphatic system?
So you mean as far as like the marker?
Exactly.
Okay, so so the question was around kind of the marker of barrier integrity for lymphatic endothelial cells. And so VE cadherin is the main one that we usually see in looking at, VE cadherin, apologies. And so that is kind of one of the major cell-cell junction molecule that will either see increase or decrease regarding the barrier integrity. You can see, and like sometimes PCAM, so that is one, but VE cadherin is definitely the major marker for that.
Okay, yeah.
The spheroid, yeah. Mm-hmm.
So the first question was, spheroids is like lesions, so that means working like the vasculature, some things that might work.
Always.
The photocross linking or stiffer tissue. Do you have any concerns about for the biophysical? I worried about like being down into how that might affect your model because actually I assume you want to test your therapeutics and how that might affect normal behavior.
Yeah. So first question was around the spheroids and the comment I made about the kind of effective drug penetration and some of the difficulties there. So that's definitely an issue, and vasculature definitely would help. So eventually doing something like a co-culture situation, including a microvasculature, is definitely something to consider. Even those, but in the body, even with, if we think about vascularized tumors, the vasculature can be quite leaky because of the tumor influences, and then still the surrounding tissue microenvironment gets very stiff, and so you still will have some of those issues. So part of it is the issue getting to the portion of the vasculature, and so you would really need to have the vasculature around the periphery, not just in that aggregate itself. And so that might be a way to think about the model, as well as kind of creating the vasculature outside of even the spheroid aggregate, so that it could facilitate into the spheroid itself. And then your second question was around the potential impact of the light influence for DNA damage. And that was part of the reason why we did try to avoid using the Ergocure as a photoinitiator, because that is one that is definitely in the UV range. We tried to use the LAP is in the 405 nanometer. The ruthenium sodium persulfate is 400 to 450 nanometers. So trying to get into that visible light range. But then also too, this is why we're trying to work on decreasing the amount of light exposure time. So when we went to those 30, 60, and 90 seconds, trying to decrease the amount of time that we do have them exposed so that we don't have that. But looking at DNA damage as an outcome will be important. Because I always tell, particularly when I'm teaching students and we talk about the work that I'm doing or others doing, that ultimately, yes, we want to kill cancer cells, but for the purposes of our research that we're doing, we need them to stay alive long enough so that we see if we can, kill them in a different way. So definitely looking at that as an outcome, looking at viability, but also whether or not we are damaging the DNA, but still keeping them viable is definitely something to pay attention to. Thank you.
Maybe we'll take, there are a couple of online questions. The first one's quite short, I think. How is the matrix stiffness measured in your studies? Is it using AFM or rheometer?
So we right now are using oscillatory shear in rheological testing. But we also recognize that is just giving us bulk information about the hydrogel. And particularly as we go through and do this photo cross-linking, the question is, are we getting a uniform distribution of cross-linking, or is there heterogeneity that is introduced by virtue of the fact that we're doing this cross-linking? And so something like AFM is something we'd like to move to be able to characterize any sort of heterogeneity that we're seeing, but also being able to ultimately associate perhaps cell behaviors with certain types of regional differences. So right now we're not doing AFM, but we're moving towards that for the future for characterization.
Great. So there's one more online question here that I think looks like a good question. Thank you so much for the great talk. You illustrated that the cancerous cells have higher stiffness than normal cells. Is there any research or work where addressing how to...
Oh, sorry, she came up.
No. Okay. Thank you for the great talk. You illustrated that the cancerous cells have higher stiffness than normal cells. Is there any research or work addressing how to specifically target cells based on their stiffness?
So whenever I was speaking not on the individual cell stiffness. I was looking at the whole microenvironment. However, to your question, I'm not actually sure if people are looking, because there is documented information about differences in like cell stiffness and cells. So I don't know if anybody is working specifically on that as a targeting mechanism. That I do not know. It sounds really cool, but I do not know if anybody is looking at something like that versus, being able to kind of look at more of a collective tissue interaction on knowing what's happening in the tissue from a stiffness standpoint and being able to perhaps maybe leverage some of that from a targeting perspective. But for the targeting of stiffer cells themselves, that I don't know about. Have to look into that.
Okay, so I think we have time for, well, we'll take these last two questions. Maybe you first, then you.
Okay.
Yes, thank you. So I know that pancreatic cancer can often involve the formation of cysts. And I was wondering if there's a way to incorporate that in your in vitro models and how that would affect overall stiffness.
So the incorporation of cysts into the pancreatic model, I haven't thought about, to be perfectly honest. I think that goes into when we're thinking about the model design and how we think about how we would model the cyst. So are we thinking about it from a cellular component, or if it's more of where you would have something like a microparticle that mimics the cyst, particularly if the interaction is more based on a biochemical interaction versus the physical interaction. So I think understanding more about that would kind of dictate how it could interact. And then that also kind of relates to if there's anything about the cyst that would be promoting any stiffening in the environment compared to, an overall situation where, you're interrupting tissue by having something else there. But I think you would get a lot of heterogeneity in terms of kind of not maybe so much bulk change to it, but definitely I would think localized changes there, but that's not something that I had considered.
So kind of going back to the question of delivery and thinking about immunotherapy, is there anything that you're aware of the influence or interaction of immune cells with the formation of this fibrosis? Like, is there any activation that occurs there?
Oh, with any interaction between immune cells and the fibrosis behaviors. Absolutely. So, one of the things that, that goes hand in hand with fibrosis is inflammation. And so when we think about all the different immune cells and how they create that inflammatory environment, it's tough whenever you're trying to create these kind of reductionist type model systems, because we know there's so many interactions going on. And so while on the one hand, we can be looking, this is part of the reason why right now our stiffening events are not cell mediated. We know that those are happening, but we're not depending on them, is so that we can have a little bit more control over it. Because outside of that, those immune cells, just like the cancer associated fibroblasts, would be part of either secreting enzymes that are part of the cross-linking process, secreting factors that will influence these stromal cells and the cancer cells to secrete matrix and things like that. So there's definite involvement, that's for sure. And so it gets very, very complex. And so right now we're trying to work a little bit independently of some of those other cellular events. One, make our lives a little bit easier without doing a co-culture situation, but so that we can exercise a little bit more control to eventually start adding those other cell populations in the models.
So I think we're out of time. Let's thank Professor Whittington.
Right. And thank you for the invitation and your time.